So next is me, so I'll just jump into it. Channeling the original volume, I'd like to start with this quote by Stebbins. Weeds of one sort, this is him describing the association between weeds and human activity at the beginning of his chapter. Weeds of one sort or another have probably existed since the middle of the Pleistocene, and the major groups of weedy species may well have virtually completed their course of evolution by the time the first great civilizations reached their climax. And I think this was the dominant view since Darwin, that evolution was this slow process of imperceptible changes. But of course, more recently, we understand evolution. We understand that evolution can occur very quickly over contemporary timescales. And this is the phenomenon that I'm interested in the context of global change and the effects that humans have on the environment. Because these human activities represent changes in the environment experienced by natural populations, which imposes selection, and the ability of those populations to evolve in response to natural selection can have consequences for the survival and persistence of those species. And in particular, when we think about invasive species and how selection may act on invasive species, I like to think about this as two sort of nested spatial scales. There's the sort of metapopulation scale, or large scale, particularly when we think about a lot of North American invaders, which were introduced from another continent and then spread within North America. So we have two very large native and introduced ranges, and then asking how evolution or how natural selection differs between those two very different places. And there's ideas about selection for colonization ability, changes in evolution of faster reproduction, of course, the escape from natural enemies, where some of the particularly specialist natural enemies are lost during colonization. And this changes selection acting on those populations. And of course, a number of biotic and abiotic differences that may exist between the native and introduced range for a particular species. And one important point here is that if we think about not just what selection looks like, but what the consequences of a response to selection would be, in this case, we predict some sort of evolution of a more invasive genotype. So in the case of escape from natural enemies, for example, actually, sorry, I'll come back to that in the next slide after this. I like to also think about selection, because we know selection acts on much more local scales, at least in native populations. And so there's another scale of comparisons we can make in invasive species is to think about selection varying among populations within the introduced range. And again, going back to the North American example, many of our invasive species were introduced to a relatively restricted range, and then have spread across a large part of the continent. And this large geographical area represents variation in climate and biotic factors. And so in this case, a response to selection for these local conditions doesn't necessarily produce an invasive genotype per se, but can facilitate range expansion by creating this sort of feedback loop where species colonizes, adapts to those local conditions, that increases survival and reproduction and creates more propagules for further invasion. And of course, we saw really nice examples of this kind of thing with cane toads. But the questions I'd like to address here are thinking about evolution of invasive species at these two spatial scales, and ask, are there common patterns of divergence, again, both at the large native introduced comparison level, as well as among populations within the introduced range. And then to ask, how does natural selection differ both between ranges and among introduced populations? And finally, to ask, how common is local adaptation in invasive species? And again, local adaptation we can think of both in terms of if we were to do a reciprocal transplant between continents or ranges versus among populations within the introduced range. So I'm going to focus primarily on the species that I've worked on, which is purple loosestrife. in eastern North America, where I've used a combination of common garden, reciprocal transplant experiments, and a little bit of theory, although I almost hate to use that word being in this session, because for me it seems like so much simpler than what everyone else is going to present. Also tell you about the key point here is looking at adaptive evolution during invasion of the species. But then to try to generalize, I'll quickly tell you about two meta-analyses that I've done. One is a meta-analysis of common garden experiments comparing native and introduced populations. The other one was an attempt to look at the two data sets on selection gradients. And then finally, I'll just show you a simulation that looks at founder effects when a species spread along geographical gradients. So just a bit of background on purple loosestrife. There's some controversy about exactly what constitutes an invasive species, but I think by virtually any definition, purple loosestrife can be called invasive. It's a rapid colonizer. It dominates habitats where it's found in North America, and it has strong ecological effects as a consequence. So just a bit of biology, which I thought I would frame in the context of Baker's list, which Mark Van Clunen nicely put up in his talk. So this is a plant that has rapid seedling growth. It's very simple to germinate, has no special environmental requirements. It shows very plastic growth, particularly in response to nutrients and water availability. Its inflorescence are indeterminate, so it grows vegetatively and then at some point switches to produce inflorescences and continues until environment becomes unfavorable. Contrary to Baker's list, it's tri-stylus and self-incompatible, but it is pollinated by a wide range of pollinators, and so natural populations are rarely pollinator limited, and a plant will typically produce from 1,000 up to 100,000 plants, seeds. It's also a late season flowering plant, so it doesn't have that rapid time to reproduction, and it has a limited vegetative reproduction, but it does regenerate quickly if damaged or cut in any way. Okay, so I mentioned the enemy release hypothesis earlier in the introduction. This is just the idea that species like purple loosestrife, which were introduced into North America, lost some of its special enemies, and particularly two species of beetle, which have large effects on fitness. And so the idea is that once these species are lost, once these herbivores are lost, purple loosestrife experiences a demographic release, and that might explain the success of the species in North America. And then in 1995, which is interesting that this is exactly 30 years after Baker and Stebbins, there was this very influential paper by Blasi and Notzold who took an evolutionary perspective of this idea and said, well, if these plants are producing costly defenses and that those enemies are no longer present, then selection may favor plants that no longer allocate resources to these defenses and instead grow faster or allocate more resources to reproduction. And there's a lot of controversy about this idea. To be fair, the title of the paper includes a hypothesis. And in support of this hypothesis, they showed that introduced population, I believe from New York, was twice as large as a native population from Switzerland. And one point I'd like to make is that we can use a quantitative genetics approach, classic evolutionary quantitative genetics approach, to think about this phenomenon. So going back to the enemy release hypothesis, so again, this is the multivariate breeders equation. So if we want to predict the change in a phenotype, right, evolution, we need to know something about the genetic structure of the population and the selection acting on that population. And these are just a vector of selection and a matrix of genetic variance, covariance components. And I'm sure we'll hear lots about that in Mark's talk. But the point I want to make here is there's clear predictions in this framework from these ideas. One is that selection in the native range should be stronger for defense traits than in the introduced range. And I think Jennifer talked a little bit about this in the context of herbivore presence and absence in the introduced range and how that varies. And then this idea that there's a trade-off between defense and growth in the absence of herbivores is simply a negative genetic covariance between those traits. So unfortunately, there's actually very few measurements of natural selection in this context. And so I started to think more broadly about how does natural selection vary, not just for defense and growth traits, but for a number of phenotypic traits. So I contacted Joel Kingsolver, who in 2011 combined these two data sets. These are two large data sets measuring natural selection on a number of natural populations. And in fact, in total, there's about 4,600 estimates of natural selection from 143 different studies. So I was very keen to make a comparison between native and introduced species. But as it turns out, only about 6% of these actually involves species in its introduced range. So I think this really, the main point here is that we really are lacking information on what selection looks like for introduced species, how natural populations of introduced species are under selection. So I want to tell you about an experiment that I did to look at this in purple loosestrife. These are study populations from eastern North America. So these are the Laurentian Great Lakes. Here's the Atlantic coast. And I studied 20 populations along a latitudinal gradient. But the key here is I took a population from the southern part of the range and one from the northern part of the range, and then crossed those to make segregating F2s, and then transplanted those F2s into each of 3 sites and to measure natural selection at the northern part of the range, the southern part of the range, and a site in the middle. What I found was that natural selection favors both earlier reproduction and larger size, regardless of location. And so here is a figure just showing age of maturity, or the number of days to first flower on the x-axis, and size of maturity on the y-axis in the northern site and the southern site. And here the white arrow shows the optimum phenotype. What's different about these is the rate at which fitness changes as you delay reproduction in the northern site versus the southern site. So in the north, delaying reproduction has a much stronger effect on fitness than in the southern site. So delaying reproduction in the north, where growing seasons are shorter, has a much stronger effect on your ability to produce offspring. So that's what selection looks like in the F2s. And so going back to that quantitative genetics approach that I showed you for the enemy release hypothesis, in order to predict the evolutionary response to selection, we need to know what the genetic structure of those traits looks like. And in this case, there's a strong genetic covariance between days to first flower and vegetative size, or size of the plant. And so if we project this covariance structure onto these two environments, and then imagine this experiment where we take these F2s and allow them to evolve to their optimum under the constraint that evolution occurs primarily along this major axis of covariance, it turns out that these populations are predicted to arrive at different optima, flowering early at a small size in the north and later at a large size in the south. And this genetic covariance structure is not just for these two traits, but for a whole host of different life history traits that I looked at in purple loosestrife. So one prediction is that if the fitness surface changes from this kind of thing in the north to this in the south, then the mean, or the optimum, should shift towards later, larger plants. Larger plants that flower later. And that's exactly what we see in common garden experiments. That here's latitude on the x-axis, so going from long seasons to short seasons in the north, days to first flower, and vegetative length. And you can see that there's these very strong negative correlations. And in fact, northern populations flower about 30 days earlier, but at only about half the size of southern populations. And these clines are very consistent year to year and across environments in both the greenhouse and the field. And just to visually show you this difference, here is a northern genotype and a southern genotype, both from North America. You'll recall, well, so in here I've highlighted in yellow the inflorescence length. So these pictures are all taken at the same time. Here's a diploid from the native range in Israel. And you can see that it actually took several months before this actually started to flower. And here's my 3-year-old son in lieu of a meter stick. So you can get a sense of the scale here. So these are huge differences among populations that seem to be driven by local adaptation to climate. And this made me think back to those early studies by Blase and Nossel. Now we're approaching the 20th anniversary. It's kind of a nice time to revisit that, to think about the consequences of this local adaptation for making these large-scale comparisons between the native and introduced range, given that we've found a doubling in size within North America. Thanks. So this is a meta-analysis I did on common garden comparisons, where studies take populations from the native range and the introduced range, grow them in a common garden, and look for changes in phenotype, genetically based changes, because it's a common garden environment. And then I asked, what happens if those populations that are sampled, so each of these points is a population, if they run along a particular environmental or geographical gradient, what happens if, how does local adaptation to local environment affect that? So here's the trait value, which could be anything like flowering time or size. And if the trait that we're looking at is random with respect to that gradient, then we recover the true difference. So here we're showing the average for all the introduced populations in red and the average for all the native populations in green. And you can see there's no difference. But if those populations fall along a gradient, if they form a cline, then if we don't take into account this cline in the analysis, we end up with different means, even though it's just populations adapting or responding to those local environments, rather than any inherent difference between the native and introduced range. I just was curious about whether this was actually having an effect in these studies. So I did a meta-analysis just looking at effect size for different combinations of traits, different classes of traits. So all traits, a principal component of all traits, size-related traits, reproductive traits, or defense traits, and then asked what happens if we ignore latitude in the analysis, and what happens if we include latitude in the analysis. And the point is not which traits are different. The point is you get a very different answer depending on whether you include latitude in the analysis. But the reason I'm hesitant to draw any conclusions about these is that most studies use very few populations. And so this data suggests that clines are probably common in introduced species, even though most studies haven't been designed to test for that. But then I was curious about whether these clines are necessarily adaptive. In other words, if we see a latitudinal cline or a geographical cline, can we infer that these populations are locally adapted? So I just did a very simple simulation for a quantitative trait. So this is many loci, each with additive effects, simple quantitative model of a diploid with 2 alleles per locus in Hardy-Weinberg equilibrium. And the key point here is that there's absolutely no selection on these loci. And what happens if a population, if a species spreads by subsampling from an initial inoculum and founding new populations, but fast enough that these populations aren't reaching any kind of equilibrium, they're just, spread is just a subsampling effect. And what you can see here is for a number of founder sizes and loci, that on average, there's no change in the phenotypic value for any of these simulations. But you can see individual cases where you get very strong clines. So these darker lines just show the extreme, the top and lower, upper and lower 5% of cases. But it turns out if you just do a linear regression of each of those 1,000 simulations, about 2/3 of them show a significant latitudinal cline, even though there's absolutely no selection in this model. So this is just the effect of founder effects and drift in this linear expansion. And I'm sure we'll hear more about this. Russ talked a little bit about fixation of deleterious alleles and how a process like this can actually increase the frequency of those. So it's a very similar idea. So then the question is purple loosestrife locally adapted? We know from natural selection measurements that the natural selection in those environments predict that those clines are adaptive. But I wanted to actually test this doing a reciprocal transplant experiment using 150 individuals from 3 pairs of populations in the south, mid-latitude, and north, grown at the same locations. And this is a study done over 4 years. And that basically the bottom line is, yes, populations are locally adapted. And the way that local adaptation arises is that the northern genotypes flower early and then senesce for the winter. But they do the same thing regardless of environment. So they have relatively constant fitness across the northern, mid-latitude, and southern site. But the southern genotype, which delays reproduction and then gets larger to produce more offspring, is just starting to reproduce when these environmental conditions become unfavorable. And so it gets hammered as it moves north. So not only is local adaptation, not only are these populations locally adapted as predicted by the measurements of natural selection, but the specific pattern of local adaptation where there's a relatively flat reaction norm for the north and a steeper reaction norm for the south is exactly what's predicted by that. So just to conclude, population divergence is common among introduced species, among introduced populations, and this creates challenges for making native and introduced comparisons. How common is adaptive evolution? There's really few studies of natural selection in invasive or introduced species, and we really need more field studies to test adaptation and measure natural selection. I was going to end with this quote, but maybe I'll just skip ahead. And I just want to advertise that in 2015, I'll be starting a faculty position at Queen's. So if anyone knows of graduate students, postdocs who'd be interested in working with me, I'd be very happy to meet them. So thank you.